So here we will discuss on the first pathway, that is classical pathway of complement activation. So this is like a cascade reaction. We have several steps in this complement activation. You can actually write this as a flow chart and draw at least three figures from the figures that I am going to show now. So the first step is we should talk about C1Q. The C1Q is actually a enzymatic system. So it consists of C1Q R2S2, means it will have one moiety of Q, two moieties of R2 and two moieties of S. Okay.
Continuation.
So this is totally C1QR2S2. We can see the diagram here. C1Q is this one, and in this C1R2S2 is like the ring, the green color ring which you can see over C1Q. So basically first what happens is C1R, one component of R, one moiety of R, it undergoes autocatalysis, and this is going to activate the second C1R. Okay, so the first C1R which has underwent autocatalysis and the second C1R which has got activated by the first C1R, these two are going to activate C1S. Let us read the sentence. C1Q binds antigen bound antibody. So this is the antigen and these are the antibodies, the green ones. So these antibodies are now bound by C1Q. So which means C1Q R2S2 totally. So now R C1R undergoes autocatalysis, activating the other C1R. We have two R C1Rs here, remember that. So that C1R2, both R's are going to activate C1S. Okay, now the activated C1S is going to act on two more complement compounds, that is C4 and also C2. C4 is cleaved into two fragments. The larger fragment we call it as C4B and the smaller fragment C4A. In this, the larger fragment C4B is going to go and bind on the antigen. This is antigen surface. It binds on the antigen surface. C1S also cleaves C2. It cleaves C2 into a larger component which we call as C2A, is the larger component here, and the smaller fragment is C2B. So again in this C2 also the larger fragment C2A binds onto the antigen. Now from C4B component, from C2 C2A component, both the larger fragments were bound on the antigenic surface. Now this total thing C4B and C2A together we call it as C3 convertase. When I say C3 convertase, we understand that it is going to convert other complement component, that is C3. Okay, so yes, this C3 convertase, this total thing we said it is called as C3 convertase. So C3 convertase is going to convert C3 into, it is going to cleave C3 into a larger fragment. See, this is C3, it is cleaved into a larger fragment, that is C3B, and a smaller fragment C3A. Again, the larger fragment C3B is going to join our C3 convertase. C3 convertase we know already, it is C4B2A. Now C3b also joins here. Now this whole thing has become C4 B2A 3b. Now this whole uh, components are now referred as C5 convertase. You should remember one thing, that when the complement undergoes cleavage, always the larger fragment is referred as B and the smaller fragment is designated as A, except in the case of C2. In the case of C2, larger fragment is written as is designated as C2A and small one as C2B. Except in C2, in all cleavages, we call larger fragment as B and small as A. Exception is C2. Okay, now we understood what is meant by C5 convertase. So whenever the larger component of C3b also joins, this whole thing now we are calling it as C5 convertase. By the name C5 convertase, we understand that this is going to convert C5 component. Yes, this is going to convert C5 component into again the smaller fragment C5A and larger fragment C5B. So C5 convertase converts C5 into a larger fragment and a smaller fragment. Now this C5b, which is the larger fragment, which is cleaved by C5 convertase, it binds to other component C6. This initiates the formation of something called as membrane attack complex. Generally it is written as MAC, membrane attack complex. So here you can see that C5b is joined by C6, C7, further it is joined by C8 and finally it is also joined by C9. So we should understand that C5b, 6, 7, 8 also poly C9. All these will join together to form something called as membrane attack complex. So this membrane attack complex, it is going to form pores in the cell of bacteria or any other target cell. It is going to form a pore like structure and which will help in the leaking out of all electrolytes so that the cell will undergo lysis. Hence this is called as membrane attack complex, and this is how bacterial cell or any other target cell undergoes lysis upon activation of this complement system.